this was suggested to me a while ago, but I'm now finally getting to it, as I have a fairly exhaustive list of different videos that I'm planning on making. And with that said, if you have anything you would like me to read about on Wikipedia, please leave it in the comments below, because I read all of them, and this series of course runs on viewer suggestion. Now, we are looking at the fungus. Before we begin, please remember that I can't pronounce scientific terminology or names or anything like that, so please do not murder me for my slaughter of the English language. Anyway, on to fungus we go. A fungus, plural fungi or funguses, is any member of the group eukaryotic organisms that includes microorganisms such as yeasts and molds, as well as the more familiar mushrooms. These organisms are classified as a kingdom, which is separate from the other eukaryotic life kingdoms of plants and animals. A characteristic that placed fungi in a different kingdom from plants, bacteria, and some protists is chitin in their cell walls. Similar to animals, fungi are heterotrophs. They acquire their food by absorbing dissolved molecules, typically by secreting digestive enzymes into their environment. Fungi do not photosynthesize. Growth is their means of mobility, except for spores, a few of which are flagellated, which may travel through the air. Fungi are the principal decomposers in ecological systems. These and other differences place fungi in a group of related organisms named the Eumycota. The true fungi are Eumycetes, which share a common ancestor from a monophyletic group, an interpretation that is also strongly supported by molecular phylogenics. This fungal group is distinct from the structurally similar myxomycetes, slime molds, and oomycetes, the water molds. The discipline of biology devoted to the study of fungi is known as mycology. In the past, mycology was regarded as a branch of botany, although it is now known fungi are genetically more closely related to animals than to plants. That's a bit freaky when you think about it. Abundant worldwide, most fungi are inconspicuous because of the small size of their structures and their cryptic lifestyles in soil or on dead matter. Fungi include symbionts of plants, animals, or other fungi, and also parasites. They may become noticeable when fruiting, either as mushrooms or as molds. Fungi perform an essential role in the decomposition of organic matter and having fundamental roles in nutrient cycling and exchange in the environment. They have long been used as a direct source of human food, in the form of mushrooms and truffles as a leavening agent for bread, and in the fermentation of various food products such as wine, beer, and soy sauce. Since the 1940s, fungi have been used for the production of antibiotics, and more recently, various enzymes produced by fungi are used industrially and in detergents. Fungi are also used as biological pesticides to control weeds, plant diseases, and insect pests. Many species produce bioactive components called mycotoxins, such as alkaloids and polyketides, that are toxic to animals, including humans. The fruiting structures of a few species contain psychotropic compounds and are consumed recreationally or in traditional spiritual ceremonies. Fungi can break down losses of crops due to fungal diseases, e.g. rice blast disease, or food spoilage can have a large impact on human food supplies and local economics. The fungus kingdom encompasses an enormous diversity of taxa, with varied ecologies, life cycle strategies, and morphologies ranging from unicellular aquatic cectrids to large mushrooms. However, little is known of the true biodiversity of kingdom fungi, which has been estimated at 2.2 million to 3.8 million species. Of these, only about 120,000 have been described, with over 8,000 species known to be detrimental to plants, and at least 300 that can be pathogenic to humans. Ever since the pioneering of the 18th and 19th century taxonomical works of Carl Linnaeus, Christian Hendrik Persson, and Elias Magnus Fries, fungi have been classified according to their morphology, e.g. characteristics such as spore colour or microscopic features, or physiology. Advances in molecular genetics have opened the way for DNA analysis to be incorporated into taxonomy, which has sometimes challenged the historical groupings based on morphology and other traits. Phylogenetic studies published in the last decade have helped reshape the classification within kingdom fungi, which is divided into one subkingdom, several phyla, and 10 subphyla. On to etymology. The English word fungus is directly adopted from the Latin fungus, mushroom, used in the writings of Horace and Pliny. This in turn is derived from the Greek word sphongos, meaning sponge, which refers to the macroscopic structures and morphology of mushrooms and molds. The root is also used in other languages such as German Schwamm, sponge, and Schimmel, mold. The word mycology is derived from the Greek myx, meaning mushroom, and logos, meaning discourse, and also meaning a heap of other things, of course. As you would know if you watch my Stoicism videos, that logos means a lot of stuff. It denotes the scientific study of fungi. The Latin adjectival form of mycology, mycology, appeared as early as 1796 in a book on the subject by Christian Hendrik Persson. The word appeared in English as early as 1824 in a book by Robert K. Greville. In 1836, the English naturalist Miles Joseph Berkeley's publication, The English Flora of Sir James Edward Smith, Volume 5, also refers to mycology as the study of fungi. A group of all fungi present in a particular area or geographic region is known as mycobiota, plural noun, no singular, e.g. the mycobiota of Ireland. On to characteristics. Before the introduction of molecular methods for phylogenetic analysis, taxonomists considered fungi to be members of the plant kingdom because of similarities in lifestyle. Both fungi and plants are mainly immobile and have similarities in general morphology and growth habitat. Like plants, fungi often grow in soil and in the cases of mushrooms form conspicuous fruit bodies, which sometimes resemble plants such as mosses. The fungi are now considered a separate kingdom, distinct from both plants and animals, from which they appear to have diverged around 1 billion years ago, around the start of the Neoproterozoic era. Some morphological, biochemical, and genetic features are shared with other organisms, while others are unique to the fungi, clearly separating them from the other kingdoms. Some of their shared features include, with other eukaryotic eukaryotes, fungal cells contain membrane-bound nuclei with chromosomes that contain DNA, with non-coding regions called introns and coding regions called exons. Fungi have membrane-bound cytoplasmic organelles such as mitochondria, sterile-containing membranes, and ribosomes of the ATS type. They have a characteristic range of soluble carbohydrates and storage compounds, including sugar alcohols, e.g. mannitol, disaccharides, e.g. trehalose, and polysaccharides, e.g. glycogen, which is also found in animals. With animals, fungi lack chloroplasts and are heterotrophic organisms, and so require performed organic compounds as energy sources. With plants, fungi have a cell wall and vacuoles. They reproduce by both sexual and asexual means, and like basal plant groups such as ferns and mosses, produce spores. Similar to mosses and algae, fungi typically have haploid nuclei. Euglenoids and bacteria, higher fungi, euglenoids, and some bacteria produce the amino acid L-lysine in specific biosynthesis steps called the A-aminodebate pathway. The cells of most fungi grow as tubular, elongated, and thread-like filamentous structures called hyphae, which contain multiple nuclei and extended by growing at their tips. Each tip contains a set of aggregated vesicles, cellular structures consisting of proteins, lipids, and other organic molecules, called the Spitzenkoppe. Both fungi and oomites grow as filamentous hyphal cells. In contrast, similar-looking organisms such as philatmus green algae grow by repeated cell division within a chain of cells. There are also single-celled fungi, yeasts, that do not form hyphae, and some fungi have both hyphal and yeast forms. In common with some plant and animal species, more than 70 fungal species display bioluminescence. Onto unique features. Some species grow as unicellular yeasts that reproduce by budding or fission. Dimorphic fungi can switch between a yeast phase and a hyphal phase in response to environmental conditions. The fungal cell wall is composed of glucans and chitin. While glucans are also found in plants and chitin in the exoskeletons of arthropods, fungi are the only organism that combine these two structures and molecules in their cell wall. Unlike those of plants and oomycetes, fungal cell walls do not contain cellulose. Most fungi lack an efficient system for the long-distance transport of water and nutrients such as the xylem and phloem in many plants. To overcome this limitation, some fungi, such as the armillaria, form rhizomorphs, which resemble and perform functions similar to the roots of plants. As eukaryotes, fungi possess a biosynthetic pathway for producing terpenes that use melaphonic acid and pyrophosphate as chemical building blocks. Plants and some other organisms have an additional terpene biosynthesis pathway in their chloroplasts, a structure fungi and animals do not have. Fungi produce several secondary metabolites that are similar or identical in structure to those made by plants. Many of the plant and fungal enzymes that make these compounds differ from each other in sequence and other characteristics, which indicates separate origins and convergent evolution of the enzymes in the fungi and plants. On to diversity. Fungi have a worldwide distribution and grow in a wide range of habitats, including extreme environments such as deserts or areas with high salt concentrations or ionizing radiation, as well as in deep sea sediments. Some can survive the intense UV and cosmic radiation encountered during space travel. Most grow in terrestrial environments, though several species live partly or solely in aquatic habitats such as the chitin fungus, Bacterium dendrobitis, a parasite that has been responsible for a worldwide decline in amphibian populations. This organism spends part of its life as a motile zoospore, enabling it to propel itself through water and enter its amphibian host. Other examples of aquatic fungi include those living in hydrothermal areas of the ocean. Around 120,000 species of fungi have been described by taxonomists, but the global biodiversity of the fungus kingdom is not fully understood. A 2017 estimate suggests that there may be between 2.2 and 3.8 million species. In mycology, species have historically been distinguished by a variety of methods and concepts. Classification based on morphological characteristics such as size and shape of spores or fruiting structures has traditionally dominated fungal taxonomy. Species may also be distinguished by their biochemical and physiological characteristics, such as their ability to metabolize certain biochemicals or their reaction to chemical tests. The biological species concept discriminates species based on their ability to mate. The application of molecular tools such as DNA sequencing and phylogenetic analysis to study diversity has greatly enhanced the resolution and added robustness to estimates of genetic diversity within various taxonomic groups. Now we move to mycology. Mycology is the branch of biology concerned with the systematic study of fungi, including their genetic and biochemical properties, their taxonomy, and their use to humans as a source of medicine, food, and psychotropic substances consumed for religious purposes, as well as their dangers such as poisoning or infection. The field of phytopathology, the study of plant diseases, is closely related because many plant pathogens are fungi. The use of fungi by humans dates back to prehistory. Otzi the Iceman, a well-preserved mummy of a 5,300-year-old Neolithic man found frozen in the Austrian Alps, carried 2 species of polypore mushrooms that may have been used as tinder or for medicinal purposes. I was actually lucky enough to study Otzi the Iceman in high school, and I'm pretty sure that there's a lot of concurrent debate about whether or not the plants were there for medicinal purposes or as kindling, but the most suggested hypothesis is that they were used for both, because it seems unlikely that he would have those specific fungi with him, seeing that they did have antibiotic properties, so they would be able to help you stop getting a local area infection if you applied it, which is crazy to think that we had antibiotics 5,300 years ago. Or was it 5,300? Yeah, 5,300 years ago. But its material and shape would also be good as tinder. So I think most people that debate about it now say it probably was both, seeing as Otzi the Iceman also had some other very advanced things on him, such as, his metal axe that he had, which, was during the Stone Age. So there's proof that they did know some things, of course. Anyway, that's a sidetrack. Maybe we'll read Otzi's Wikipedia one day. Ancient peoples have used fungi as food sources, often unknowingly, for millennia in the preparation of leavened bread and fermented juices. Some of the old and written records contain references to the destruction of crops that were probably caused by pathogenic fungi. On to history. Mycology is a relatively new science that became systematic after the development of the microscope in the 17th century. Although fungal spores were first observed by Giambattista della Porta in 1588, the seminal work in the development of mycology is considered to be the publication of Pier Antonio Micheli in 1729's work Nova Plantarum Genera. Micheli not only observed spores, but also showed that, under the proper conditions, they could be induced into growing into the same species of fungi from which they originated, extending the use of binomial systems of nomenclature introduced by Carl Linnaeus in his Species Plantarum. In 1753, the Dutch Christian Hendrik Persson, from 1761 to 1836, established the first classification of mushrooms with such skill as to be considered a founder of modern mycology. Later, Elias Magnus Fries, from 1794 to 1878, further elaborated the classification of fungi, using spore colour and microscopic characteristics, methods still used by taxonomists today. Other notable early contributors to mycology in the 17th to 19th century and early 20th centuries include Miles Joseph Berkeley, August Carl Joseph Quodder, Anton da Berry, the brothers Louis René and Charles Delaunay, Arthur H.R. Buller, Curtis G. Lloyd, and Pierre Andrea Saccardo. The 20th century has seen a modernization of mycology that has come from advances in biochemistry, genetics, molecular biology, and biotechnology. The use of DNA sequencing technologies and phylogenetic analysis has provided new insights into fungal relationships and biodiversity, and has challenged traditional morphology-based groupings in fungal taxonomy. Now we move to morphology, starting with microscopic structures. Most fungi grow as hyphae, which are cylindrical thread-like structures from 2 to 10 in diameter and up to several centimeters in length. Hyphae grow at their tips, apices. New hyphae are typically formed by the emergence of new tips along existing hyphae by a process called branching. So they grow and split and then branch out and continue growing. Or occasionally growing hyphal tips fork, giving rise to two parallel growing hyphae. Hyphae also sometimes fuse when they come into contact, a process called hyphal fusion or anastomosis. I think a good example of this is if you watch, those microscopic or even normal camera views of fungus growing at speeds that are, of course, sped up. And, of course, at the micro level, you'll be able to see what I'm talking about. These growth processes lead to the development of a mycelium, an interconnected network of hyphae. Hyphae can be either septate or coenzytic. Septate hyphae are divided into compartments separated by cross walls, which are internal cell walls called septa that are formed at right angles to the cell wall, giving the hyphae its shape, with each compartment containing one or more nuclei. Coniotypic hyphae are not compartmentalized. Septa have pores that allow cytoplasm, organelles, and sometimes nuclei to pass through. An example is the dolipore septum in fungi of the phylum Eumycota. Coenzytic hyphae are in essence multinucleate. supercells. Many species have developed specialized hyphal structures for nutrient uptake from living hosts. Examples include Hostoria in plant parasitic species of most fungal phyla, and arbuscules of several mycorrhiza fungi which penetrate into the host cells to consume nutrients. Although fungi are opisthokonts, a grouping of evolutionary related organisms broadly characterized by a single posterior flagellum, all phyla except for the chatrids have lost their posterior flagella. Fungi are unusual among the eukaryotes in having a cell wall that, in addition to glucans and other typical components, also contains the polymer chitin. Onto macroscopic structures. Fungal mycelia can become visible to the naked eye, for example on various surfaces and substrates such as damp walls and spoiled food, where they are commonly called molds. Mycelia grown on solid agar media in laboratory petri dishes are usually referred to as colonies. These colonies can exhibit growth shapes and colours due to spores or pigmentation that can be used as diagnostic features in the identification of species or groups. Some individual fungal colonies can reach extraordinary dimensions and ages, as in the case of a colonial colony of Armillaria solipes, which extends over an area of more than 900 hectares, which is 3.5 square miles, with an estimated age of nearly 9,000 years. The apitheum, a specialized structure important in sexual reproduction in the Asimites, is a cup-shaped fruit body that is often macroscopic and holds the hymeneum, a layer of tissue containing the spore-bearing cells. The fruit bodies of the Basidiomycetes, the Basidocarps, and some Asimomites can sometimes grow very large, and many are well known as mushrooms. The growth of fungi as hyphae or in other solid substrates or as single cells in aquatic environments is shaped for the efficient extraction of nutrients because these growth forms have high surface area to volume ratios. Hyphae are specifically adapted for growth on solid surfaces and to invade substrates and tissues. They can exert large penetrative mechanical forces. For example, many plant pathogens, including Magniporthia grisera, form a structure called an aposium that evolved to puncture plant tissues. The pressure generated by the apressorium directed against the epidermis can exceed 8 megapascals, which is 1,200 psi. The filamentous fungus Bacillomyces lineaceus uses a similar structure to penetrate the eggs of nematodes. The mechanical pressure exerted by the apressorium is generated from physiological processes that include intracellular turgor by producing osmonoids such as glycerol. Adaptations such as these are complemented by hydrolytic enzymes secreted into the environment to digest large organic molecules such as polysaccharides, proteins and lipids into smaller molecules that may then be absorbed as nutrients. The vast majority of filamentous fungi grow in a polar fashion, extending in one direction. Other forms of fungi growth include intercalary extension, which is longitudinal expansion of hyphal compartments that are below the apex, or growth by volume expansion during the development of mushroom stipes and other large organs. Growth of fungi as multicellular structures consisting of somatic and reproductive cells, a feature independently evolved in the animals and plants. Wow! Has several functions, including the development solely on carbon fixed by other organisms for metabolism. Fungi have evolved a high degree of metabolic versatility that allows them to use a diverse range of organic substrates for growth, including simple compounds such as nitrate, ammonia, acetate, or ethanol. In some species, the pigment melanin may play a role in extracting energy from ionizing radiation, such as gamma radiation. This form of radiotrophic growth has been described for only a few species. The effects on growth rates are small, and the underlying biophysical and biochemical processes are not yet well known. This process might bear similarity to CO2 fixation via visible light, but instead uses ionizing radiation as a source of energy. Okay friends, that ends the first part of the interesting fungi Wikipedia reading. The next section we're going to be talking about fungal reproduction, so stick around for that. I hope that you're excited